Have you ever wondered what keeps a living cell from just spilling out? It's this, the cell membrane. And if you think it's just some simple wall, you've got to think again. It's a gatekeeper, it's a communicator, and it's pretty much the skin of life itself. So let's dive in and see what really makes it tick.
I mean, just look at this incredible thing. This is not a static, rigid wall. It's this bustling, crowded, constantly shifting environment. You see all those different molecules kind of floating in there? They're not just stuck in place. They're on the move all the time. So yeah, let's completely ditch the idea of a simple container and really explore this dynamic living barrier.
Right. So we're really reframing how we think about this. The membrane isn't just a passive bag holding all the cellular goo inside. No, it's so much more like a cell's skin. It's always sensing what's going on around it, it's talking to other cells, and it's being incredibly selective about what gets in and what stays out.
And that really gets us to the heart of the matter, doesn't it? It's the big question for this whole explainer. How can something that's so incredibly thin and seems so fragile do all these complex jobs? How is it both a barrier and a gateway at the exact same time? Well, the answer lies in one single, pretty remarkable molecule that makes up the very fabric of this living barrier.
And here it is. This is the hero of our story, the phospholipid. This tiny little molecule has a huge secret, a secret that lets it build these massive, self-organizing structures all on its own, with zero instructions from the cell. This guy. This is the key to everything.
So, that secret? It all comes down to a property called amphipathic. Now, that's a fancy word, I know, but it stands for a really simple and incredibly powerful idea. This molecule has a split personality when it comes to water. One part of it absolutely loves water, but the other part is totally terrified of it. And this push and pull, this love-hate relationship, is the driving force behind the entire cell membrane.
And this shows that split personality perfectly. You've got the round, hydrophilic head group. It's totally happy to be surrounded by water. But then you have these two long, hydrophilic fatty acid tails, and they will do anything to get away from water. It's this constant tug of war that makes the whole structure of the cell membrane possible.
So, okay, we've got these amazing two-faced molecules. The next question is, how do they go from just being a bunch of individual molecules to building a massive, complex thing like a membrane? The answer is kind of mind-blowing. They do it all by themselves. It's like magic. It's a process we call self-assembly.
Just think about it for a second. What's going to happen when you dump millions of these things into the watery environment inside a cell? It's almost instinctual, right? The water-loving heads are going to turn towards the water, and the water-fearing tails will desperately try to hide. That simple, unavoidable reaction is what drives the formation of the entire membrane.
But you know, there's one more piece to this self-assembly puzzle, and that's just basic geometry. As you can see here, not all phospholipids are shaped exactly the same. Some are kind of cone-shaped with smaller heads, while others are more like a cylinder. And this tiny little difference in shape, it has a huge impact on the final structure they can build together.
And boom, here's the result. Those cone-shaped lipids, they huddle together to form a little ball called a micelle, with all their tails safely hidden on the inside. But the cylinder-shaped ones, the ones we really care about today, they form this magnificent structure, the phospholipid bilayer. It's two layers of molecules, with all the tails tucked away from the water right in the middle. It's a perfect, self-sealing barrier.
So here's the crucial point. This bilayer thing isn't something the cell has to work hard to build piece by piece. Nope. It's just the most natural, most stable way for these molecules to arrange themselves in water. It's a self-assembling, self-healing barrier. It is, without a doubt, one of nature's most brilliant and efficient designs.
Okay, so we've built our wall. But remember what we said at the beginning. It's not a brick wall. It's much, much more like Jell-O, right? It has structure, but the individual pieces, in our case, the phospholipids and proteins, are totally free to float around inside it. This property is called fluidity, and it's absolutely critical for the membrane to do its job. And the secret to this Jell-O-like fluidity often comes down to a simple little bend, a kink in the tail. See that bend in the molecule on the right? That's caused by a double bond between two carbon atoms, which makes it an unsaturated fatty acid. That permanent bend is like having an elbow sticking out, and it stops the tails from packing together too neatly. This comparison makes it super clear. Saturated fats, they have these straight tails like a neat stack of logs. They can pack together really tightly, which creates a more rigid, less fluid membrane. But those unsaturated tails with the kinks in them, they create space, kind of like a jumbled pile of bent straws, which makes the membrane way more fluid and flexible.
But what happens when things get too hot or too cold? Well, that's where this little molecule, cholesterol, plays this really important and kind of weird role. It wedges itself right in between the phospholipids and acts like a fluidity buffer. When it's hot, it holds things together, stopping the membrane from turning into a soupy mess. And when it's cold, it gets in the way of packing, keeping it from freezing solid. It's like the membrane's own personal thermostat.
All right, so we've got this fluid, jello-like structure, but what does that actually mean for the molecules living inside it? Well, it is definitely not a calm, placid sea. It's way more like a molecular mosh pit, just this constant, chaotic motion. Everything is in a state of perpetual dance.
And look, this isn't just me coming up with a fun metaphor. It's a core scientific observation. The very fact that these lipid molecules can move around and diffuse so freely within their own layer is literally the definition of a two-dimensional fluid. It's a sea of molecules, always shifting, always rearranging itself.
Now this shows us a really fascinating detail about our molecular mosh pit. Not all dance moves are created equal. Molecules can zip around sideways, that's lateral diffusion, at incredible speeds. But for a molecule to actually flip-flop from one side of the bilayer to the other, well, that's a huge, energy-intensive process. It's like trying to do a somersault in a packed elevator. It just doesn't happen very often on its own.
And this chart really drives that difference home, doesn't it? The bar for lateral diffusion is a skyscraper, while the bar for the flip-flop is, what, a speed bump? This means each layer of the membrane is basically its own distinct neighborhood. Molecules mostly stay on their own side of the street. You've got two separate, bustling communities just living back to back.
Okay, this all sounds great in theory, but how can we be so sure? How can you prove that this molecular dance is actually happening inside a living cell? Can you watch it? The answer is yes, you totally can with a really clever and elegant technique.
It's called FRAP. That stands for fluorescence recovery after photobleaching. I know, it sounds super complicated, but the idea behind it is simple and just brilliant. First, scientists label the proteins in the membrane with the fluorescent dye, so they all glow. Then they take a powerful laser and zap, they blast a tiny spot, which basically turns off the lights on the molecules in that one area. And then they just watch.
And what they see is just incredible. Over time, that dark, bleached-out spot slowly starts to glow again. Why? Because unbleached proteins from all around just drift in and fill the gap. This is the aha moment. It's the direct, visual proof that everything in the membrane is in constant fluid motion. It's like watching a tiny glowing river start to flow again.
So let's just quickly recap this whole incredible journey. We started with that amazing two-faced phospholipid. We saw how its love-hate relationship with water forces it to spontaneously self-assemble into a perfect bilayer. We learned that this bilayer isn't a rigid wall at all, but a fluid, jello-like structure. And finally, we saw the definitive proof that this fluidity allows for a constant, chaotic, and beautiful dance of molecules.
The big takeaway here is that this perfectly tuned fluidity isn't just some neat biological trick. It is absolutely essential for the cell to function, for proteins to meet each other, for signals to get sent. Which leaves us with one last and really important question. What happens to a cell, or even to us, when this perfect, delicate balance gets thrown off? What are the consequences when the cell's living barrier starts to break down?